Okay, so thanks to Adam for the chance to tell you about a lot of very new ideas, and certainly I think this is an example of, like any other perspective from aging, a parochial perspective, in this case, very much from C. elegans, but it's kind of the idea is to try to move towards the general, and it's also very much about looking at aging at a higher level than the molecular in this particular talk. So I work at the Institute of Healthy Aging, and I guess in a way, the ultimate goal of the work is to improve human lifelong health and well-being. And we've had a bit of good news lately in terms of that direction. I think the changes in the government in America probably will be helpful in that respect, not least our US rejoining the Paris Climate Agreement, and of course the vaccinations news is absolutely wonderful. But in terms of lifelong health and well-being, I think really here are some of the biggest enemies to human well-being. These are actually the leading causes of death in the developed world today. And you can see here, let me put on this, I'll put on the laser pointer. So we can see for the predominant causes, cardiovascular disease, cancer, respiratory disorders, that'll be particularly COPD, nervous system disorders. A lot of that will be things like dementia, digestive disorders, all the way down. So in terms of what's causing these causes of death, they are predominantly caused by the aging process. So here we have a major biological process, which is now the leading cause of death and disease, serious disease in the world. And yet it's a biological process that we actually don't understand. So this is a tremendous challenge in terms of medical research. Probably, I would say, the most important question in biomedical research is to understand the main cause of disease in the world. So why has science been unsuccessful in understanding aging as it has been? And I think the simple answer is that aging is clearly a very complex and multifactorial process. So this is a good job for a simple, tractable model organism like C. elegans. Very, very simple and tractable. So if one were able to use the sort of simplicity of this organism to be able to understand aging, and the fact that the lifespan is only a couple of weeks is very, very convenient. It has wonderful genetics and so on and so on. If we could understand C. elegans aging, I think this would really be a major landmark in terms of bringing us closer to being able to understand aging in an organism that actually matters, namely our own species. So a lot of what's been happening in model organism genetics during the last 30 years is the use of genetics to understand aging. So lifespan, we know from looking at the different lifespans of different species, is primarily a process that's under genetic control. It's influenced by the environment, but very strongly genetically controlled. So given that it's a genetically determined process, one possible approach to understand aging is to use mutants. So if we can find mutants with altered lifespan, then it should be actually technically quite simple to discover the genes and the proteins that are controlling lifespan, and the identity of those gene products should really could provide some sort of explanation for what aging is. And the real excitement came in the late 80s and early 90s when it was discovered by Mike Klass and Tom Johnson and Cynthia Kenyon that you really can get long-lived mutants in C. elegans. Single gene mutations can increase lifespan. This is from a classic paper from Cynthia Kenyon's lab. You can see here there's a survival curve with the percent of the worms alive against time. So here, these white squares, these are wild-type normal worms. And these black squares, this is a population of worms grown at the same time. And they've got a single base change in this gene called DAF2, which is increasing lifespan. So this is an incredible result. And I was blown away by this as I was a graduate student at the time. I just want to kind of emphasize how mind-blowing this result is. So firstly, what it implies is that there must exist kind of upstream of all of these diseases of aging, some sort of core aging process, which is controlling the entire thing. There's actually some central aging mechanism controlling all of these different diseases of aging. It's an astounding thing. And I think it's sort of inspired and fascinated people and drawn them into the field. And it's something that it's a cause that you can also intervene in, potentially to protect against a whole spectrum of aging-related disease. It's incredible. But also, not only that, but this is in C. elegans. And C. elegans is, famously very, very simple and tractable. So it ought to be pretty simple to figure out, what's happening here. So it's amazing. So I kind of got interested in this in the early 90s, and I thought, well, how difficult can that be? I think maybe 5 years of research or something, and we'd get an answer to this basic question: what is aging? What's the nature of aging? So I moved over in the early 90s to the Don Riddle's lab. He discovered DAF2. He worked on the genetics of DAO formation. At University of Missouri, Columbia. But it wasn't 5 years. I mean, this is 1993. This was an incredibly long time ago now. Look at this. This is when John Major, you remember him? He was Prime Minister. And this is Jodie Comer here from Killing Eve. This was the year of her birth. So it's taken a very, very long time. Things did move quickly initially during the 90s. It was quickly discovered what some of these longevity genes control, and that led to the identification of this insulin IGF signaling pathway. This is extremely simplified, but essentially there's an insulin IGF receptor, DAF2 in the worm, which acts through a signaling cascade to control a transcription factor called DAF16. It's a FOXO class transcription factor. So this transcription factor is somehow controlling the aging process in the worm. Okay, so then things carried on moving quickly. I moved back to the UK, and I was lucky enough to be able to collaborate with some labs at UCL, Sally Levers and Linda Partridge and Dominic Withers, to look at this pathway's role in aging in Drosophila in the mouse. And in fact, it turns out that if you reduce signaling through this pathway, you also see a delay in aging of a smaller magnitude in flies and mice. So this is an evolutionarily conserved function of this pathway to control aging. But the big question, the really fascinating question is, yes, so what's aging? And the problem is DAF16, it's a transcription factor. It's controlling many, many genes downstream. And I think the field kind of hit the buffers around in the early half of the noughties. And it hasn't really progressed very far since then. This is a long time. So we still don't know what DAF16 is controlling. So this is, these genes were sequenced 20 years ago. And there's been by now thousands of papers published on C. elegans aging, and yet you still don't really have an account of what aging is in fundamental terms in the worm. So about 10 years ago, this really started to worry me, and I thought, if we're not getting the answers, that's telling us that there's something fundamentally wrong in our approach. It was getting increasingly dull listening to these endless molecular talks, with never a satisfying explanation. So I think I decided that it must be that we're using the wrong experimental approaches, and we're probably thinking in the wrong way about aging. So what I'm going to talk about today is a whole bunch of very new approaches and new concepts that have emerged, particularly from my own lab in the last years. So here's our problem. I'm going to talk first about experimental approaches. So here's the problem. We've got these regulatory proteins like DAF2 and DAF16, these control pathways, and they're working through some sort of downstream effectors, of which there are thousands, intractably so. And then somehow this is leading eventually to death at a different time. So the question is, what's the chain of events happening in between these downstream genes and death? So here's what I think is happening. It's not really very controversial what I'm saying here, actually. What I think is that the reason why the worms die of old age is because they get sick. They get diseases that limit life. And so these diseases will be caused by some form of senescent pathology, some sort of deterioration, disease causes, and then those senescent pathologies will be generated by some pathogenetic mechanisms. And so in the worm field, we have thousands of papers about sort of genes controlling lifespan, tons and tons of knowledge, but there's actually very little knowledge about this whole end of this causal chain. So we decided, I've spent sort of 10-15 years kind of working on this end, I was going to move to the other end. So what are these various steps? Each one of them involves interesting questions in their own right, but actually the big question in terms of understanding aging is, what are the pathogenetic mechanisms underlying senescent pathologies? That's, I think, the key, and it's sort of not where people have been looking previously. So this is what I call a pathology-centered approach, as opposed to thinking about explanations for aging in terms of molecular events, more looking at the pathologies of aging. So this is, again, looking at higher order type of information. So let me start by saying a little bit, because I've covered a lot of ground in the last years. It's been actually a wonderful period. It's a lot of kind of radically new stuff. So I've done some work. We've done some work on the question of what are the causes of death in the worm, and also the biology of death itself. The actual process of organismal death is another kind of blind spot in the whole process, very little studied. So talking about causes of death, what do the worms die of? This was actually years ago. George Williams, I met in when I was at Missouri. He asked me, what are all worms die of? And that's a very challenging question, one that people haven't really asked much. But I did some work with an excellent postdoc called Yuan Zhao, and she did necropsies on worms that have died. Nobody had done this before. So you take the corpses of the worm and then you examine them. It's like an autopsy, a necropsy, because it's an animal. And what she found, there were two different types of corpse in C. elegans. You get ones with a swollen pharynx, and you get other ones that die later with a shrunken pharynx. So they actually, this aging worm population have two different types of death. So they're dying of different things. And if you take the survival curve and you actually separate the deaths from, we call this a big P because it's got a swollen pharynx, and we call this a small p. It's got a little pharynx. You can separate them out. And so you have, here are the small p deaths, here are the big P deaths. And it turns out a lot of the treatments that affect lifespan in worms, they give you these strange survival curve shapes. And it's because they're having differential effects on big P death and small p death. So this helps us to understand the shapes of survival curves, and actually focuses understanding of gene action on aging to actually each specific form of death. So that's sort of a helpful way to start to break down survival curves into individual causes of death. Again, very briefly, I want to just mention this other aspect, which is a completely unexplored area, or very unexplored area in biology, which is what actually happens during death. There's actually a very complex biology taking place during the death process. Like human beings, when they die after you declare dead legally, there's a whole lot of biology that continues for a long period afterwards. It's very interesting. So in C. elegans, we've actually been studying the process of organismal death. Actually, I'm just going to skip over this because it's sort of, but just to give you the gist of this, the worms actually experience something which is equivalent to mammalian rigor mortis, which shows how there are common mechanisms of organismal death that occur between mammals and even down to nematodes. I'm not going to bother to go into this actually, but just to mention that, sort of to kind of sort of slightly cheaply emphasize that this is a cool upcoming area. There's actually a new journal coming out called Death Research. I'm honored to be on the editorial board. So it's all about the biology of death. So I'm going to skip over that for time. Because I think what's more interesting is to think about theories of aging, because I think here is where the worm field has been held back, and probably the broader field. So here's our problem. What is this control? What is this pathway that has these huge effects on lifespan? What's the aging process downstream? So one way to kind of approach this for developing hypotheses is to look at theory. What do we think aging is? What do people believe it is? There are huge numbers of different theories of aging, but quite a lot of them share a basic sort of idea, which is, I call it the damage maintenance paradigm, where you start off life at the end of development in a youthful state, and then molecular damage accumulates, and then you arrive in a senescent state and get diseases and so on. And the rate of damage accumulation is actually can be controlled by maintenance functions like detoxification, repair, turnover of damage, and so on. Okay, so what I'm going to do now, I'm going to jump over an enormous amount of stuff which I've previously been talking about for years, and I hope you don't mind this. But I spent 10 years sort of testing oxidative damage theory in C. elegans with constantly negative results. And I think that it's clear that damage type theories are not adequate for explaining aging in C. elegans. They simply haven't succeeded in explaining what's going on in these mutants. So I don't want to go into all of the detail there, but lots and lots of tests of the oxidative damage theory have turned up negative. And so it's difficult ever to completely exclude the theory, but what I've done instead is to look at alternative theories and see if they do better. And the short of it is that they very much do in C. elegans. So I'm going now into a realm of alternative theories to the damage theory, using as a starting point a very well-known and very well-accepted theory in aging, which is certainly not a reductionist theory, which is the evolutionary theory of antagonistic pleiotropial trade-offs, which was introduced by Williams. So the evolutionary argument is that aging is not an adaptation. It reflects the fact that natural selection force declines with increasing age. And what Williams argued was that because genes, new alleles of genes can be very pleiotropic, having many effects at different tissues, different times, you have many genes will have effects across the life history. And if they have benefits early in life, but also cause pathology later in life, they might actually still be selected for. They might overall increase fitness. So you argue that aging evolves as a side effect of natural selection in favor of mutations that cause a benefit during youth. But the Williams theory doesn't say that much about, what are the actual proximate mechanisms of aging. But one thing that it does say, which is useful in terms of thinking about kind of causes of aging, is that he's arguing that the main cause of aging is actually the action of wild-type genes. And I think he's absolutely right about that. So he's arguing that it's primarily a wild-type trait. It's not a trait which is purely caused by sort of accumulation of harm. It's actually caused by genes. And in the case of C. elegans, you've got DAF2, the wild-type DAF2 gene is, of course, shortening lifespan dramatically. So this is certainly the case that wild-type DAF2 is causing aging. But what are the proximate mechanisms? So the sort of the popular theory to try to link the sort of evolutionary, the ultimate theory with the proximate is the disposable soma theory from Kirkwood. So he assumed that aging is caused by damage, molecular damage. He reasoned that maintenance is costly, and so he suggested that what natural selection does is to optimize resource investment so that the maximum amount of resources can go into reproduction and just enough resources go into maintenance to keep the animal alive during reproduction. So very elegant theory. So what results is what he calls a disposable soma. It's a sort of throwaway soma. So it's one of these theories which is very beautiful, but it simply hasn't been supported empirically. It was put forward in the late 70s, and the evidence is just not there that this is true, however elegant it is. But I think it's the right sort of theory. It's trying to link the evolutionary and the proximate. So there's this evolutionary proximate kind of theory. There are alternatives to disposable soma, which actually go back to Williams himself, who did speculate very briefly about how antagonistic pleiotropy might work in terms of actual mechanisms. And he used this kind of totally fake example. He imagined this example where you've got a gene for calcium deposition. So you have a gain of function in the gene. And so that means that, this might be a gazelle in the Serengeti. The bones grow faster and it can run away from cheetahs, and that enhances fitness. But then that same gene in later life might promote calcium deposition in arteries and promote cardiovascular disease. So this is not disposable soma. This is something quite different. This is the gene actually actively causing pathology. And this idea was picked up on and developed in terms of, to explain a lot of the recent findings by a guy called Mikhail Misha Blagasklony in the States. And he was thinking similarly to Williams, but thinking in terms of regulatory gene products rather than structural ones, like targeted rapamycin, IGF-1, insulin, IGF signaling in the worm. These are all growth promotion pathways. That's what they primarily do, is to promote growth. So he argues that what you have here, you get early life fitness here, not from a simple function, but from entire developmental, reproductive, reparative programs involving hundreds, thousands of genes. And then those programs then become futile programs in later, what he calls quasi-programs, to drive the development of pathologies. So this is approximate ultimate theory, alternative to disposable soma theory. Blagasklony doesn't argue that damage doesn't contribute to aging. And what he does argue is that the aging field is obsessed with molecular damage to the exclusion of all other possibilities. And that actually these pathways that have been studied so extensively, they probably don't work through damage. They're working in a different way. And so he comes up with a term which I think is helpful, which is to say, well, the argument here, this is very broad brush, that aging is not so much a damage process like a passive type of process. It's actually an active process. It's actually actively driven by genes. So it's not loss of function that's causing it. It's actually the opposite. It's actually what he calls hyperfunction. And that's turned out to be a useful term. So just to emphasize the difference between Williams' ideas and Blagasklony, because people sometimes say, oh yeah, Blagasklony is just Williams. It's nothing new. Blagasklony's ideas are a development of Williams's. Williams, as I said, was clearly thinking in terms of structural genes, where you have lots of genes, individual genes having very small effects alone. So here, from Williams's scheme, you wouldn't expect something like DAF2 with a large effect on aging. Whereas Blagasklony is seeing how you can have core growth pathways actually promoting a whole range of different developmental programs in different tissues, and then those run on to become these quasi-programs. So quasi-program, this means the non-adaptive run-on of a developmental program in later life. And this doesn't necessarily mean development itself, but also other kinds of complex program. And that could include things, for example, like inflammaging, this chronic switching on of sterile inflammation in later life. And I think that what I find very elegant about this theory is that these pathways like insulin, nitrous signaling, TOR, they're driving diverse programs and quasi-programs. And that can explain how you can get single genes with large effects on lifespan. So I think it's interesting to kind of think historically here and look back to around about the year 2000. So in 2000, we already knew about these pathways like the growth hormone, IGF-1, targeted rapamycin, and so on, that they have these effects on aging from worms up to rodents, and evidence slightly for humans as well. So we've done the genetics, we've found the genes, but what does that tell us about the mechanism? So at the time we thought, well, this is a pathway that controls growth, but it also is involved to some extent in somatic maintenance. So the assumption was, well, somatic maintenance, that's damage, that's aging. So obviously growth has nothing to do with aging. So that's just a sort of a byproduct. But what the growth and development part is also possibly even more important than somatic maintenance in terms of the control of aging. So in a way, I think this is an example of where science was kind of blinkered by its preconceptions about the phenomena that it was studying. So I'm going to sort of rest of the talk. I'm going to talk about. So I'm sort of wondering about my time, and we started late. So what? Sort of 20 past. Okay. All right. I'm doing all right. So my lab has been kind of exploring how well this Williams-Blagasklony theory, how well it performs in terms of generating hypotheses that we can test that turn out to be right. So I'm just going to turn off my heater. And in fact, it's done incredibly well. I spent years getting negative results with damage theory tests, but with this quasi-program tests, they've turned out really well. So we've looked at a range of different senescent pathologies, diseases of aging in the worm. Worms have their own weird diseases: intestinal atrophy, yolk steatosis, uterine tumors, disintegration of gonad. And we've been able to show how we can explain these pathologies, at least to some extent, in terms of as resulting from quasi. So here's an example of Williams-Blagoslawny theory and how it can account for senescent pathology in C. elegans. So this is delving into details of the diseases of aging of worms. So here you can see the front end of a healthy young adult worm. And if you look at the cursor here, I'm showing you here the intestine, which is a large structure that also functions as the adipose tissue and also the liver of the worm. It's a kind of multi-purpose organ, very large in the worm. Now here's middle-aged C. elegans, and you can see two different pathologies here. Actually, look at the bottom first. This is here's the intestine of an older worm, and you can see that most of the biomass of the intestine is gone. It's atrophied to a dramatic degree. So the red line here is the lumen of the gut. You can see it's massively expanded. It's turned into a sort of empty bag. And here's another pathology. These are pools of what look like olive oil in the worm. And these are lipid-rich pools that also contain vitellogenin, which is the yolk protein. Cutting a long story short, we were able to explain the origins of these pathologies as follows. So here we have a cartoon of a young worm and a cross-section of the intestine. These yellow spots are yolk granules in the gut. So the gut is the site of synthesis of yolk. Yolk's transported across the body cavity into the oocytes to support the growth of embryos. So what happens as the worms get older is that the intestine actually consumes its own biomass in order to maintain the production of yolk. Even though these worms are post-reproductive, they carry on producing yolk in what has been long assumed to be a sort of futile biosynthetic activity. And this vitellogenesis is strongly promoted by insulin nitrous signaling. So this is also an autophagy-dependent process. So it's eating its own biomass to make yolk. So the run-on, the futile continuation of this process leads to the atrophy of the intestine and the appearance of these sort of pointless pools of yolk in the body cavity. So we were really excited about this because what we have here, finally, we've got an actual process which is an etiology of senescence, which not only does it instantiate the Williams-Blagoslawny model, it's a quasi-program, it's a futile vitellogenic program, but it's also promoted by DAF16 and by DAF2 and suppressed by DAF16. So it's an actual process of senescence downstream of DAF16. So I spent, 15 years trying to figure out what's going on downstream of DAF16 causing aging. So we were very excited about that. But what I'm going to talk about for the rest of the talk is another twist in the story, which is very new. We've just, most of this is available on bioRxiv and preprints.org when it's submitted, but it's not published yet. So this is findings which I think are really shocking and actually really change our whole understanding of C. elegans aging and even aging more broadly. So I'm excited to talk to you about this. So just talking with a graduate student who arrived some years ago, we were exploring the idea that perhaps this yolk production in post-reproductive worms, could it somehow be an adaptation? It's a sort of niggling doubt always that is it really futile? But how could it be useful to a worm to produce yolk when it's not reproducing? But Karina Cairn came up with a possible explanation, which is that the C. elegans, once they finish laying eggs, they actually vent yolk onto the plates that we keep them on through the vulva. So this is a worm where the yolk has been tagged with green fluorescent protein. And you can see here a pool of yolk just in the, it's in the uterus, and then it's come out here in a pulse. And if you look on plates where the worms have been, you can see these pools of yolk on the surface. So we then thought, well, maybe the larvae actually consume the yolk. Maybe the yolk is serving a function. So I'm going to show you here a little movie where you can see the yolk-venting behavior. Let's get it going there. Okay, there we go. So here's the vulva here, and you can see the pool of yolk in the, here it goes. So you see a little pulse of yolk. So that's, there it goes again. So that's yolk venting. So this is a mother who's finished laying eggs and is now venting yolk. So Karina characterized the yolk venting. This is age of worms. This is egg laying here in black, and then this is yolk venting. So you can see there's a pulse of yolk venting immediately after the end of egg laying. So here's the critical test of this hypothesis, which is to see whether vented yolk can support larval growth or have any effect on larval growth. So the experiment was to take mothers who've stopped laying eggs and are just starting to vent yolk, put them on plates with no other food source, so no E. coli for the worms to grow on, and allow them to vent, and then take the mothers away, and then put worm eggs onto those plates, and then look at see whether the presence of the yolk is actually affecting larval growth. So that's what we see here on the y-axis. This is the length of the larvae, the baby worms. So here we have initially. Let me get the laser pointer again. So here we have control worms, and here are plates with yolk on them. So okay, so the larvae are growing more, but this could be because of secretions from the worm or some other thing apart from the yolk. So here we have another test where we have as a control, we have larvae on the plates, preconditioned plates with larvae, and here preconditioned with venting mothers. So again, you see increased larval growth. But I think the sort of killer experiment was this one, where what we did was to take mothers where we'd knocked out yolk synthesis, and we treated plates with mothers either with the yolk synthesis knocked out or normal mothers, and you can see that there's a difference. So knocking out yolk synthesis was blocking the advantage. So it's the yolk which is actually supporting larval growth. This final experiment on the right, this was a kind of a cute test where we took mothers who were just laying their last eggs on a plate, and then we either left the mothers to vent or we took the mothers away. So this is seeing whether mothers' vented yolk can actually support the growth of their own larvae. And you see a benefit to leaving the mothers there. The larvae grow faster. We also tried replacing the mothers with surrogate mothers which had the yolk synthesis knocked out. And again, you lose the growth advantage. So what this is telling us is that this vented yolk is actually serving a function which is equivalent to that of a milk. And so we gave a nickname to this vented yolk. We called it yolk milk. So this yolk milk venting is promoted by insulin signaling, which is this pathway which, if you remember, accelerates aging. So here you can see this is looking at the vented yolk levels in wild type. So this is normalized to 1. And here are 2 DAF2 mutants. They don't vent at all. So insulin signaling is promoting lactation in the worm. Here we've got mutants which have got elevated insulin signaling. So for example, this is a P10 mutant with elevated PI3 kinase signaling. And you can see they've got elevated yolk venting. So here are the conclusions. And there are some sort of kind of cute and interesting conclusions, but there are also some really freaky conclusions. So the cute conclusion is that the worms exhibit lactation. So this might sound kind of really crazy. I initially thought so, but then I learned that in fact, there are a number of invertebrates that actually do produce milk to feed their offspring, including the tsetse fly and the Pacific beetle cockroach. But in terms of our papers that we published just recently, what this is implying is that this later yolk production, it's not actually futile at all. So our interpretation of this being a quasi-program is actually incorrect. In fact, it's something more like a costly program which is functioning as part of a trade-off between intestinal atrophy and increased fitness through increased larval growth. But it gets weirder. This type of phenomenon where you have this sort of large-scale remobilizing of resources to support reproduction, leading to destruction of somatic organs, this is not something that's very typical of aging in most animal species, but it is something that you see in organisms which are semiparous and show something called reproductive death. So this is organisms that reproduce once and make a massive suicidal reproductive effort. I'll call this RD for short for convenience. And that includes things like the Pacific salmon. Famously, they swim upriver and then die in the upper reaches of rivers after having spawned. This is a marsupial, Antechinus stuati, where the males will undergo they go into a sort of reproductive frenzy and then die within a couple of weeks. Now, this is where it gets really weird. So it's been known for a very long time that these kinds of semiparous organisms, if you actually block this reproductive death, which you can do in various ways, like by gonadectomy, of course, you see huge increases in lifespan. And this raises the question of whether, in fact, the increases in lifespan that we see in C. elegans are actually the result of suppression of reproductive death. And in fact, we have a paper which is out in bioRxiv where we've shown evidence that ablation of the germline, which extends lifespan and suppresses reproductive death in C. elegans, is actually really that this is how it's working. It is specifically a suppression of reproductive death. So this is kind of alarming, because what this is, on the face of it, what it's saying is that this wonderful extension of lifespan that we're seeing in C. elegans, this is not something to do with suppression of aging, it's to do with suppression of reproductive death. So this is the scary thing, not the milk production. So what does this mean? This has huge implications. So just to reiterate here, there are two basic types of life history in animals and plants. You have iteroparous life histories, which are more common in mammals, in animals, where you can have multiple rounds of reproduction, like in Drosophila and mouse in humans. Semiparous, you have a single burst of reproduction, which can be often suicidal, followed by rapid death. So we have proposed a sort of an understanding, which I think is well supported by evidence through literature searching and so on, which actually argues that although C. elegans die from reproductive death, it's still useful for understanding aging in iteroparous species. And that's based on the hypothesis that reproductive death is actually a single element of iteroparous aging that's been amplified. So kind of in the past, there's been a tendency to view iteroparous and semiparous organisms as completely different. But semiparous species evolved from iteroparous species, and one can find actually species that kind of all the way from extremely iteroparous to extremely semiparous, with everything in between. So you have, in terms of aging, what you could consider an etiological continuum. So, for example, in mammals, during lactation, there is something which one could clearly describe as a costly program, where bone is actually consumed, it's actually eroded in order to release calcium to support milk production. So this is repurposing, costly repurposing of calcium for milk production. But in mammals, from mice up to humans, you get lactation associated osteoporosis. In fact, women who are lactating are vulnerable to bone fracture. But at the end of lactation, the bone actually regrows. So it's a reversible cost. But in something like lactation in C. elegans, and yolk production is, in a way, it's the ancestor of milk production in terms of evolutionary history. What you have here is you have a costly program where it's actually an irreversible cost. It's a terminal investment leading to reproductive death. So by that view, then what we have is one mechanism of aging which is operative in higher animals, but which is greatly exaggerated in C. elegans and becomes kind of the predominant cause in C. elegans, which in a way is a good thing, because it means we can study that particular element of aging in relative isolation. So here's the hypothesis that we put forward. This is again, it's another preprint. We can't submit it until we've got several other results papers into press. So what we're arguing is that it's something like this. This is a slightly unusual representation here. So here's age on the x-axis. The y-axis here is a measure of the how lethal a given pathology is. So here we have an aging organism with a range of different senescent pathologies with different etiologies, okay? So this is based on an assumption that aging is a multifactorial process where multiple etiologies are working together. I think it doesn't do to say, you know, ROS causes aging or quasi-programs cause aging. They are kind of types of etiology that work together. I think this is probably why aging has been so very difficult to figure out, because there are multiple etiologies operative. So what's happening in a semmelparous organism is that these programmatic type of etiologies, like the quasi-programs, costly programs, and so on, they've been greatly exaggerated, and they cause massive pathology long before the other types of etiology come into play. So if, for example, in a Pacific salmon, you can gonadectomize them, and that you get a massive increase in lifespan. So instead of dying of reproductive death, they kind of die of aging in the normal sense. You're removing these programmatic etiologies, and then they'll die of a range of other etiologies. In an iteroparous organism, everything is, all of these etiologies are all acting together. And that's why if you look at, for example, many species where you reduce these etiologies, you do see effects on lifespan, but they're much smaller than those that you see in C. elegans. And in humans, it's not clear that they're having much of a role at all. Actually, or at least their contributions are relatively small. So that's a sort of a very new perspective on understanding the relationship between reproductive death and aging, and the way that different classes of etiology are contributing to aging in different types of organism. So I'm kind of drawing to a conclusion. I just wanted to mention we put out the milk paper, the milk-venting paper, as a preprint, and actually it already got noticed. Even there was a thing in New Scientist about it, but it hasn't been peer reviewed. And there's so much wonderful stuff that appears on the internet. And this was one of my favorites. Microscopic worms pee milk on their children as their bodies decompose. So I think I can see the error here, because you think of a sort of pee coming out the vulva, you know, but worms actually pee out of a little hole in the side of their head. So I guess Brittany Borovich didn't realize that, so she kind of drew the wrong conclusions there. All right, so I want to end sort of trying to make some global sort of description of what I've been talking about here. So this is kind of, again, kind of trying to move towards the big perspective on aging. So this is, in this slide, a kind of history of the last 25 years, certainly of my life and many other labs. So we've got this evolutionarily conserved pathway controlling aging across many species. And how people have been pursuing this is with this assumption that aging is caused by molecular damage. So this pathway must somehow be controlling somatic domains. And that might include, for example, promoting proteostasis, depending on where the failures of proteostasis are coming from during aging. So what we've recently been arguing in papers we published over the last few years is that actually what's happening downstream is something else is happening, which is that we have these programmatic mechanisms, particularly futile run-on and quasi-programs, and these are causing multiple senescent pathologies. And by this view, actually, we don't have a single aging process. We're already moving towards multiple mechanisms of aging. That's part of the Williams-Blagoscloni model. It's not saying that damage doesn't contribute to aging. So the idea here is that these large effects on lifespan are because of these quasi-programs affecting multiple senescent pathologies. That's why you see effects on lifespan, not because the whole aging process has been changed. But what I'm now saying is from the new work, and I've told you about reproductive death. I haven't mentioned adaptive death. We've actually published a series of papers on this over the last couple of years. This is a whole other thing, which is very, very peculiar to C. elegans and a few other species like budding yeast, definitely not applicable to other organisms. So we're now saying that this reproductive death and adaptive death, these are things that are likely not happening in Iteroparus species. But this doesn't seem to make sense by itself because well, first of all, obviously this raises concerns about C. elegans as a valid model for studying aging in higher animals. But remember that, as I told you, these pathways do have effects on lifespan in Iteroparus species. So how can we get this circle to square? So here's how I think we can do it. So arguably what we see in C. elegans, the way C. elegans behaves, is very much as if there is some central aging process causing all of its diseases of aging. But in Iteroparus organisms, in higher organisms, the picture really doesn't seem to hold. It seems much more plausible that what you've really got is multiple different types of pathogenetic mechanisms, but some of them are actually, if you like, etiologies of multimorbidity. And that would include things like these programmatic etiologies driven by insulin IGF signaling and mTOR. Okay, so this I think is kind of like an optical illusion, if you like, particularly from the results from the worm, which are kind of not fully representative of what's happening in higher animals. And remember that the effects on lifespan are much greater proportionally in C. elegans than in any of these other organisms. So here we have DAF16, which is suppressing this process. But what we've been arguing is that DAF16 is actually suppressing reproductive and adaptive death. That's why we're getting these huge effects on lifespan in the worm. So, you know, what does this tell us? In a way, kind of going back to the beginning of the talk and kind of thinking myself, kind of going over to Missouri and so thrilled by discovering, you know, the possibility of discovering this central mechanism of aging. It reminds me a little bit of when I was a little boy. You know, I was crazy about superhero comics, you know, like Batman and Fantastic Four and that sort of thing. And I can still remember when I was very small, when I realized that actually Batman and Superman and so on actually didn't really exist. And I can still remember the feeling of disappointment, you know, realizing that, you know, Batman actually is not real. It was sad. But actually, I think these extraordinary implications from these discoveries from Johnson and Klass and Kenyon, you know, actually, it's kind of not real either. There aren't these core mechanisms that control the entire aging process. So that's really sad. But on the other hand, I think it's really exciting that actually, you know, in the end, if you have knowledge, if you have understanding, you have no idea where this is going to take you, but it's very powerful. What we have, I think the field is most likely showing is that we have these etiologies of senescent multimorbidity, which are a huge opportunity in terms of future medical interventions. You know, and I think that one of the causes of these types of major etiologies of multimorbidity are these programmatic type of mechanisms like quasi-programs and costly programs. So I think, you know, we can still learn a great deal about aging from C. elegans, and I think it does illustrate how a fundamental understanding of aging in any organism is going to, in a way, it creates a starting point for a broader understanding of aging. So my last slide here, this is actually how I view aging at the moment. It's a sort of it's an evolving process of developing ideas, which is really what my lab is trying to do, is to sort of develop theories and ideas about aging in general from the worm. So, okay, so here's the premise, or kind of, I think, a reasonable guess, if you like. Across the animal kingdom, you get this huge diversity of pathologies of aging, diseases of aging, within any species, enormously diverse. Human diseases worm diseases. They look very different to one another across species and even within species, but plausibly the fundamental causes in broad brush terms are not going to be entirely different. And I think in a way the most plausible view is that there are actually a limited number of basic principles of senescent pathophysiology which are operative across the animal kingdom. But for any particular pathology and for any particular species, the particular etiologies that are operative will vary. Okay, so here's just, this is very, very sort of hypothetical. So here we've got, this isn't a claim that this is how it is, but this is the sort of thing that's going on. We've got a number of basic causes of disease. So infection, I mean, that's germ theory of disease. That's a fundamental principle. In different organisms, you have different degrees to which these are important. So for example, programmatic mechanisms, and that's programmatic antagonistic pleiotropy, is hugely important in C. elegans. Whereas, for example, DNA damage appears to play, it seems to be important in mammalian aging, but appears to play no role in C. elegans. So again, this is, you can see, if this is how aging is, you can see how very difficult it's been to pick it apart. So the point is that by studying something like C. elegans and by studying its weird and different diseases that it has, we can learn about these fundamental conserved principles of senescent pathophysiology, and identifying those and understanding how they combine with each other to give rise to different diseases of aging. I think that's the key to being able to understand aging as a process, and the route to being able to develop powerful explanations for all of these diseases that we don't understand, like Alzheimer's disease, like osteoporosis. We don't understand them at the moment. So, okay, that's all. I hope that sort of gives a very broad brush overview of the sort of things we do and some of these very new ideas. Just to say that I've just been incredibly lucky these in terms of the people that I've got to my lab, and that, you know, these ideas, and these are in contrast to, you know, the disposable soma theory or the free radical theory or the error catastrophe theory. Those were theories that were dreamt up by people sitting in armchairs. These theories are derived from experimental research on C. elegans and from extensive literature research on data coming from the field of experimental gerontology. So these ideas are much more empirically based, but a lot of it has come from people working on these things in my lab, and particularly, I would say, Karina Kern, who's done this amazing work on milk venting, and Marina Escura, who did a lot of the work on the intestinal atrophy pathology.